Hello, I'm Meredith Blackwell, and I'm here at the Mycological Society of America meeting in Minneapolis, Minnesota, and we're working on interviews for this oral history of mycology project that we've been doing now. This is the second season for it. And I'm going to interview Jason Stajich, and most of the time today he will interview himself. And so he's going to start talking and let us know about his life and the very good career that he's had for a young squirt. So,
Awesome.
yeah, I've known you since you were at Duke in grad school, and I thought you were on the faculty. But anyway, let's start by saying where you were born and how you grew up, you know.
Sure. Let's see. So I was born in North Carolina in Chapel Hill,
Oh,
and my parents had moved there for my dad to go to school to be a physician assistant, and so my mom was in graduate school in French Lit, and then we grew up in North Carolina, and I went to high school at the School of Science and Mathematics in Durham, which is a residential school for my last
Right.
2 years of high school. That was fun and intense, but sort of always been interested in science through. My dad worked in genetics, so I was always kind of paying attention to the way that they were studying disease in human and following families, and so I guess I kind of just sort of stayed interested in that. I went to college at Duke University for my undergraduate, but I focused on computer science, and so I was started to really get interested in how we deal with data, right? And that was kind of what I was learning as an undergrad, and then I worked in the hospital between college and grad school in a genetics program or genetics department that's actually the same group that my dad had worked in, but in a different part. So I kind of really learned about how to use these programming skills towards analyzing data, processing data, and I just got hooked on that. And then I didn't know what I wanted to do in graduate school, except I wanted to do more, basically genomics at that point, right? Processing and thinking about data. Started graduate school, rotated a couple different labs, and ended up in a lab of Fred Dietrich, who had worked on some of the early days of the first sequences in yeast, especially. So he had been part of that. So there are all these different data sets that are starting to be made available through different sequencing projects. We were doing our own, and so I worked on that at first, and then I realized it was a great system to work on, and I was like, I should probably learn what these things are. I mean, I knew what fungi were, but I didn't really know.
Yeah.
And so, because of the great community at Duke, people like Rita Sfoglius and Francois Lutoni, I could go and learn more natural history and kind of got hooked on that. At the same time, I was in a department that was working on infectious disease and looking at fungi like Cryptococcus, and so folks like Joe Heitman, John Perfect, others were there working on those questions.
Was Fred in the med school?
It was in the med school. It's a med school genetics
Okay.
department.
Yeah, I didn't realize he was not in the department.
He was on the school in biology, although I think there's a lot of cross appointments.
Yeah.
And I was part of this there was a university program in genetics and genomics, and so it was really nice because we had funding for the first 2 years of school, and so it was really intended that you had some freedom. You didn't have to work directly on a project that your thesis advisor had right away, I think. And so I actually spent a lot of time in the biology department working with friends who were developing methods in population genetics, and so I was kind of learning I learned a lot of my skills or training on that through kind of just working with those other students and faculty. So I don't know, I guess, so maybe that's why I seemed like I was already independent, because I really felt like for me the best thing was to kind of learn from different people and work with them under different questions.
Yeah, I remember the first time I encountered you was at the DPIPA meeting, and although I was PI on the project, somebody else arranged it, and here you are speaking. So you were a young squirt.
That was an honor. It was good that somebody thought to invite me, and
Yeah.
it was fun to be part of. Part of that.
We included lots of students and postdocs, and as it ended up, it was a very good program for mycology, I think. Yeah. So you finished up with Fred.
I finished up with Fred in 2006, and I had applied for a fellowship for a postdoc at UC Berkeley through the Miller program. and the Miller Institute for Basic Research, and I applied to work with John Taylor. So he, of course, had to nominate me, but also you had to get through the committee, and it was very, very competitive. Now, a previous Duke student who had been in Reedus's lab, Ann Pringle, also had a Miller Fellowship. So I had reached out to her and asked her for advice. And so, again, this is maybe a theme you may see in some of these interviews, just how much kind of mentoring and co-mentoring that you get through this broader community. And so I think right at that point, I was already getting a benefit from others that had come before. But so I went to Berkeley on that fellowship and worked with John for 3 years. On some different projects, coccidioides, the first genome of the Batrachochytrium fungus had come out, and so we were working on that and a few other projects. So.
Yeah, and with the cocci, you started showing that it was more diversified than.
Yeah. With cocci, that was fun because it was John's lab had shown there were 2 different species, but we wanted to understand a little more about what that meant at the genomic level, and also what made cocci different from its relatives. There's a specialized pathogen, and so we started comparing. There weren't. I mean, a lot more genomes now, but there were quite a few then, and we could try to ask differences between plant saprotrophs and these human pathogens or animal pathogens, and so that was a fun chance to kind of be at the forefront of asking those questions.
Right. And then you got a job very quickly.
Yeah, I got a job. It was at UC Riverside, and again, these are sort of funny stories, but also how somebody looks out for you. So I hadn't applied to the search when it was open. It was a bioinformatics search, and I maybe wasn't even quite sure I was applying that year for jobs. This was it was my second year of a postdoc, and the woman, Natasha Reichel, who had been the chair of the committee, had been at Berkeley giving a seminar. She was meeting with John, and she mentioned that this job, they were still looking for people to apply, and he told her about me, and she's like, send your CV in right away. We'll make sure the committee still gets a chance to look at it before they close it. They liked my CV, and I came down for an interview, and I got a job. It was crazy.
They needed you.
They needed me, and I felt really, really special that it could work out. So I started.
Bioinformatics jobs were hard to fill.
It was hard, and it was also because I think a lot of us. I mean, it's still kind of a new science, and so the number of people that have trained in it is still. There weren't that many. I mean,
So they were lucky. Yeah. And so what are you doing there now? You've started up your lab. It's well established.
Yeah, it's almost 10 years now, scarily. So I started my lab, and I worked on a couple of different projects at the beginning. Some of what we do end up doing gets driven by where the funding comes in, and I have a lot of different interests, but we worked partly on trying to develop databases to make working with genomes easier. So we gotten some money through the Sloan Foundation to work to support this built environment.
I just heard from Paula today.
Oh, yes.
Apparently there's a big paper that's come out, a report that was funded by Sloan in the household fungi area, and it was about looking at Legionella.
Oh, okay.
And so it looks like we'll be able to figure out how to control it and stop it from spreading in environments. Yeah.
That's great. Well, so it was fun to be part of a little bit of that in terms of the informatics around how we look at the microbes that are inside built environments. And it wasn't actually my total expertise right away. I hadn't done a lot of molecular ecology, but I really used that as a launching point to think about those questions, and we tried to build some infrastructure to help the community. And at the same time, we started applying for other grants, and the big one that has been kind of sustaining has been this genealogy of life grants that worked with Joey Spadafora and Tim James, Robbie Robertson, Carrie O'Donnell, Matt Smith, lots of really great mycologists.
And Ann's on that too, is Ann?
Ann's not part of that one. I think that's might be something different.
Okay, different one.
But this was to look at the zygomycete fungi diversity.
Oh, okay, zygolite.
So some of your and including Basidiobulus, one of your favorites, and so that has been a good chance to learn about the systematics and how we can use these genomic approaches as well as integrating fossil data and other things.
I was very disappointed. I was hoping that Lobosporangium would turn up in some of the sampling you were doing. It used to be called Echinosporangium.
Right.
It's only been found 3 times within a 2-year period by 3 different people. and never since.
You know, I've seen, and we sequenced 2 of those isolates in terms of
Yeah,
the ones
that's all
the
there
ones
are.
that were collected. But wasn't this from like soil behind a shed in or
Well,
something? Or
no, it
were
wasn't
you-
a shed. It was
okay.
a biology building.
Biology building, okay.
At Texas.
At Texas, okay.
And I was told I could go out and get Cunningamella out of the dust.
Uh-huh.
And instead, I get Lobosporangium.
Well,
And
maybe we should try harder again.
George Carroll's, I never could isolate it there again. I
Mm.
tried and tried. George Carroll's came from Nevada and the desert, I
Okay,
think.
okay.
And one more was collected, but no, there was no culture, and it came from Sonora in the desert.
Okay.
So desert environment.
Yeah, well, so that's one of the things we've transitioned to in the lab is we've got a set of projects looking at biological crusts in the desert and trying to understand the fungi. And so we know a lot about the algae, or a lot's been done on the algae and the cyanobacteria, but we're interested in what some of the fungi do in those environments. And we know, of course, about lichens that form these lichenized crusts, but there are other fungi and so that are there. And so several of my students have been working on amplicon as well as culture-based studies.
Yeah.
So we might know better. I think we need to go back. There are a few zygomycetes that show up in those
Yeah, you need to
amplicon
check
work.
those
so
out.
we need to check better to see
Yeah,
if that's
because I would
there.
love to. You know,
Yeah.
I'm sure it's there. You
Yeah.
just have to find the right little
This is
space. Yeah.
Cool. Yeah. So that's
Anyway.
kind of what we've been different ways that we've been taking on. So I think a lot of what I've decided to do has kind of pollinated with as the techniques improve or as I learn different ways that people approach these questions. And so I kind of felt brave enough to take on this more environmental microbiology question that I hadn't necessarily done a lot in before.
Yeah.
So.
Good. And so now that you're there and fairly established, how are you going after recruiting students? Is it easy?
I think a lot of students that I would say it's hard. It's hard to find students that have both skill sets in terms of a training in mycology and informatics. And so I usually have people come in and they learn informatics from me, and then
Right.
And they do better if they already have a motivation to work on something microbial. So I try to help instill that, but sometimes if they're already motivated with that, they tend to do a little bit more better there. But everybody does like a wet project and a dry project in my lab, so there's something that involves growing something or doing experiments in the lab or in the field, and then there's data analysis and computational, and some students and postdocs are more suited towards one or the other type of project, but everybody gets a chance to see how are the data collected, and maybe that's what you're really good at, but also how do you analyze it yourself?
Right.
So
Right.
that's. I mean, I think it works. Sometimes I think I could do better on how I set up the project so that we learn faster where the holes are in their training. But I think that's in the end, I just try to give people a chance to hopefully find a passion in some part of that work,
Yeah.
and then we just try to give them a place to do it.
The only way to do it is to give them a chance, I think. Yeah. Yeah, so anything else important to say?
In terms of
Your
mycological history,
career or your history or
well,
anything?
I mean, I think when you look back, I think lots of people, I feel lucky, so lucky that so many things kind of worked out. I feel like, luck is part of it, and then just being able to be in the right place in terms of being prepared. So I had computational skills. This was the time when these bigger data sets were coming out where those skills were needed. So I've had the chance to collaborate with so many people, and I consider that a real opportunity and a blessing to get to keep doing that. But it's what I hope is that I'm also teaching others to do that, so that more people can bring those skills to the table.
Yeah, and looking back, I know this meeting you've been very involved in honoring John.
Yeah.
Yeah, so tell us a little bit about that. What have
Sure.
you done?
Well, so we had a surprise for John Taylor first before the meeting started with many of his alumni from his lab that came back, and we had a surprise party and showed funny pictures of him and told stories.
Was he really surprised, do you think?
He was surprised.
He didn't know.
We let Delia, his wife, in on the secret to help make sure that he was at the right place at the right time, but he didn't know that we would all be there. And like Georgiana May, who's one of the organizers for the meeting, was his first graduate student, so it was really appropriate to have it here. And also because he retired, but I think it was really fun. And then at the meeting we had a symposium, and it was an honor to recognize some of the different ways that he's impacted the field. And to tell him, I always feel it's really important to tell people that you appreciate what they did, when they're
Right.
here in the moment, not to kind
Right.
of wait till later. So that was fun to have a chance to say that, to say those things to John in front of everybody, and we also endowed a fellowship for our an award in his name in MSA
For the graduate
for graduate
students.
students, best speaking, which is shared, of course, as a dual award that's also in your name, so that we can.
There are two of them given,
exactly.
so one will be mine and one will be his.
Exactly. I think that's great, and also just that it's both of you have a real strong component in your careers about communicating science, communicating what how you teach, how you explain it, and I think that's why we want to also honor it as a talk. Honoring students giving great talks is a really important way to market. So I'm just glad, I'm excited I could be part of helping that happen, and super glad that we could honor John and
Okay,
it's
and
fun.
you're talking about looking to the past, but
Yeah.
you are very young, and so how about to the
future.
future? What's going to happen? Do you have any ideas?
I think, I can see things changing in terms of how technology has changed a lot of what mycology does now, and so I think that I think that we're going to see different ways to really understand what fungi are doing in kind of short time scales. So how are they colonizing environments? So we look a lot at like distributions of fungi, which is historical in a sense, is why are they ecologically distributed in this way? But I also think that we're going to have these tools to be able to look at more carefully what are the competitions that they're interacting with. Where did they get their food source from in some complicated environment, using different kinds of isotope labeling that's being developed. Obviously, the sequencing is a real important part of that. How we look at really measuring. We have really sensitive methods now to measure where fungi are, where organisms are in the environment, and probably proteomics and metabolomics, these other kinds of technologies, are just give us another window into the functions that the fungi play.
Right.
And I think that's really, for me, what I hope we can get closer to is really understanding what are their ecological roles in some of these different environments, and is that why there's so many species, because they have to fulfill these different niches in a way, and then that leads to sort of diversification because different lineages kind of specialize. So
Right.
I hope that more light will be shed on that. At the same time, we want to describe more of the diversity that's out there, and I guess the high throughput methods are making that more, a lot more feasible.
So I want you to go out and look for zygomycetes, not just for logosporangium, but when I've looked at studies of gut organisms, you see very, very few zygomycetes, and Basidiolus should be there.
Should be there.
In
Okay.
humans as well as 50 other organisms. So
Okay.
I think somehow those zygos are being missed. Do you think?
I think so. I think some of the primers probably have a different bias, and I think to some extent they're less known to many of the people that are making those observations before, so they kind of get missed or ignored. So I think like I said, the sensitivity to techniques, but maybe having a better knowledge of what to look for.
Yeah, and a number of times I've contacted people who've done biome studies, and never do they know. When you actually ask them, they say no zygomycetes.
Okay. Well, I think you're right. I think they're there. We're not using the right approach maybe, and or maybe it just needs a little bit of tuning to that. So I think. It looks like there's some interesting places that, even in the desert, we've been looking at places where there's water accumulated, then you get insects there, then you start to see zygos in those environments, and so maybe it's patchy, and that's why some of it's
that's what probably happens. And I know the slime moths certainly are patchy because they're associated with the substrate. But Bob Gilbertson, when he first went to Arizona, applied for NSF money to study desert fungi, and he got back reviews saying there are no fungi in the desert. And he proved them wrong.
He proved him wrong. Yeah, no.
So you do the same.
Hopefully we can. That's good.
Yeah.
Those are good inspirational people to think about when you're the first time through. They don't believe you, but I think we can show it.
Thank you very much for taking the time, because I know you've been very, very busy. So
Well, thanks for doing this, Meredith.
Yeah.
It's good to be here.